I'd like to begin by acknowledging the traditional owners of the Kaurna country that I live and present to you from, and the many ancestral lands that I've worked on, which are listed below, and I pay my respects to elders past and present. I acknowledge the deep feelings of attachment and relationship of the Aboriginal people to country, and I respect and value their past, present, and ongoing connection to the land and cultural beliefs. I'm Alana, and I am working on the molecular-based systematics of Psilophagus, which is an important but quite problematic group of psyllid parasitoid wasps. Psilophagus is a drastically understudied genus that belongs to the globally huge superfamily Chalcidoidea, which has approximately 20,000 described species currently. It belongs to the family Incertidae, which has almost 4,000 described species worldwide from almost 500 genera now. The genus Psilophagus itself has approximately 250 described species worldwide, about 110 of which are described from Australia. It's fairly widely accepted, most of you would know this, that the number of described invertebrates in Australia is estimated at only about 30% of the actual number. When it comes to really tiny parasitic wasps that most people don't want to go anywhere near, let alone research, I'm guessing that this number is probably much, much lower for this group of Hymenoptera and for Hymenoptera in general. So the Psilophagus are the primary parasitoids of lerp-forming psyllids in Australia, and due to this ubiquitous relationship with eucalypts, which are everywhere, they're really abundant, they're really diverse, and they're really widespread in Australia. Of the global fauna, 110 of the described Psilophagus species are from Australia, but only 45 of those have sequence data available. And despite this Australian-centric distribution and hyperdiversity of the Psilophagus, of those 110 species and of the 45 that have sequence data, only one of those is actually an Australian species. This is a really good indication of the level of neglect of the group, and that not much has been done on it since molecular systematics kind of became a thing in science. So being purportedly host-specific, speciose and regulating of psyllid populations, the potential for using parasitoid wasps like Psilophagus for biocontrol is huge, especially where psyllids have become pests either outside of their natural range or where the host plants are of conservation concern. So the basic biology of how this control works is that a psyllid comes along, lays its eggs on a eucalypt, a psyllid nymph hatches and builds itself a little sugary kind of house called a lerp. A wasp comes along, parasitizes the nymph through that lerp, and once the wasp reaches maturity, it bursts out the body of the nymph and leaves that psyllid for dead. So within this group, my project specifically aims to utilise sequence data, morphology and host associations to delineate species and match them to the described taxa, where that's possible. I aim to describe new species which are associated with specific psyllid hosts. I aim to produce a DNA barcode library of Psilophagus that are read from those known hosts, develop molecular phylogenies for the genus using multiple genes to recognise species groups, and to examine the evolution of wasp and psyllid host relationships. I plan to develop standard and online keys using morphological and molecular criteria for rapid species identification, especially for non-professionals. And I want to make all of this information publicly accessible through avenues like the Australian Atlas of Living Australia. So to address the aims of this project, I start with quite intensive field work. So in the field, I find eucalypts which have lerps on them. I carefully examine the leaves and separate them out so that there's only one species of lerp on the leaves. They then go into Ziploc bags. It's very technical. They go into Ziploc bags with some paper towel. And then over the next few weeks, often up to 4 or 5 weeks, I care for them in the lab and I wait for the wasps and ideally the psyllids to emerge. Accurate field work, although laborious for this project, is at the absolute core of this. It's taken me to over 120 sample sites over 4 different states over the last 18 months to 2 years. It's really slow work. It's often hot and requires carrying around a lot of Ziploc bags and a lot of snacks through often questionable parts of our lovely country. Erin Fagan-Jeffries can attest to the perks of this field work. And after multiple 3-week field trips with me to some fairly strange and sometimes frightening parts of the country, Erin has definitely earned her stripes as an absolutely supreme field partner. So following the field work and what is often weeks and weeks of quite intense rearing and sorting of the specimens, I get to do the lab work. I've been really, really fortunate to be able to conduct the molecular component of my research at the Australian National Insect Collection, where I've built successful NGS libraries from yields as low as 0.1 nanogram per microliter of DNA, which is as horrible as it sounds. We use NovaSeq to sequence the mitochondrial genome, plus 4 nuclear genes and ultra-conserved elements. I've also tested this library and extraction protocol on some really old dried pin specimens. That's proving to be quite successful, but I think pursuing that, 2 to 2.5 years into my PhD might be out of the scope of what I'm doing. All of this wouldn't have happened without Dr. Vidushi Patel, who is part-time friendly witch and full-time molecular superstar. She's taught me all of my amazing wet lab skills at CSIRO in Canberra. So I've had a really enthusiastic and productive start to my PhD over the last couple of years. So all excited, this is where I thought I'd be by now. I thought I'd have all of my sequencing done. I thought I'd have all my phylogenies constructed and my morphological work kind of at least half underway. Thought I'd have keys started and I'd have a couple of manuscripts in the works. Unfortunately, COVID and bushfires, which affected most of my sample sites, means that I'm actually in fact about here. I've got about 90% of my sequencing and lab work done. Very, very little of the morphological work, but we do have preliminary phylogenies which are underway. And preliminary phylogenies, after a couple of years of just doing field work and basically collecting things and putting them in the fridge, are really, really exciting. These phylogenies were made possible by another incredible person who is heavily involved with my project, Dr. Juanita Rodriguez. She built these prelim trees for me while also very patiently teaching the bioinformatics necessary to analyze my sequences. Don't be too critical of them. They're still really incomplete. They need outgroups. They require so much more analysis. But it's still really cool to be able to share some early results. So the first tree we've pulled out, this is a concatenated mitochondrial gene tree. It's worth pointing out that there's never been a phylogeny constructed for Silophagus, so this is really new and really, really cool. Even the few chalcid phylogenies, which are amazing. There's some incredible people working on Chalcidoidea at the moment who have produced these incredible phylogenies, but even most of those that include the family Incertidae are quite minimally sampled and they never include Australian data. So what I'm doing is filling quite a gap in inserted systematics, which is really cool. Like I said, I need so much more time to analyze these trees. Things like conferences keep getting in the way, but already I can see there's some really cool things that are starting to come out. So there's instances like this where I've got a single female morphospecies and multiple male morphospecies that have come out of the same species, or at least the same genus of lerp, which look the same, on exactly the same tree, which are collected at the same time, but the males fall out as being completely different species. So whether the males are far more generalist, I don't know, but that's one of the things that is kind of repeatedly coming out in the tree. In instances like this, these are all morphologically similar wasps, which come out of the same looking species or the same genus of lerp from across 3 different states. So there seems to be some indication that geography and climate are going to have quite a lot to do with the speciation of these groups. So that's pretty exciting as well. Now, this baby's only a few days old, so no one asked me any complicated questions about it because I don't think Juanita's here to help me answer them. But what I've got here essentially is a really well-supported UCE tree. This is from the same data set that shows kind of some similar patterns of weird host stuff going on. But what this does do, I think, is help to resolve those species groups a lot better. There's still some really weird things going on in this, which I'll talk about later, but it's probably due to me making some early errors. So this is a concatenated single petition tree from about 1800 UCE loci. So I've done a really quick, dodgy comparison of the 2 trees, like in the last few hours. And what I'm kind of gathering from this is the difference in the number of species groups that are falling out. And I'm not going to go into too much detail about the species numbers, but if I'd color coded all of them, you'd see there's quite a bit of movement where some of them fall out. So depending how we end up delineating the species will kind of inform a lot of this comparison. So the mitochondrial tree recovers about 30 species and the UCE tree between 20 and 23, again, depending how we end up delineating them. There's something weird going on with 137, which is the one at the top with a really long branch length. In the UCE tree, it's really long and it falls out by itself. In the mito tree, it falls out really tightly within a species group with about 8 other samples, I think. So I started to wonder if I need to start thinking about whether there's any level of introgression happening with some of these species, or the more likely scenario where I've just kind of made some kind of mistake. And either way, I need to do a fair bit more work. So all of this is really amazing and new. And it's really cool and really informative if you like inserteds and wasps, but why is it important for anyone else to know all of this? Essentially, because there's no unifying character or synapomorphy for this genus, so it makes identifying them morphologically almost impossible. What this means is it makes it almost impossible for non-experts to identify them, which means that it's really hard to make inferences about biocontrol or conservation or morphological identification. Their cryptic appearance and traditional taxonomy-based descriptions means that classification is constantly in flux as we learn new things about them and we can start to do molecular work on the group. And that most definitely needs sorting out phylogenetically. Part of that means that there's real uncertainty about levels of host specificity and hyperparasitism, because there's never been a level of work that's had accurate host records associated with this. All of the Silophagus that have been described haven't been reared, so it's really hard to know which wasp actually belongs to which psyllid. There's been no focus on the global fauna, so we really need to get the Australian species revised, a new species discovered and described, so that we can get that information out there. This is where I started running out of time and just chucked some slides together, so bear with me. But the next,
you know, it's 3:45, so.
Oh my goodness. Okay, that's totally fine, because these are all my dodgy slides. Basically, I need to do more lab work. And then the rest of it was just like plugging some things that I'm going to do, including insect investigators, which Erin, you're going to talk about at the end. So that's me, done.